Great. Hey, everybody. Welcome. So glad that you are here. And I am really excited to be having this conversation with our panel and with all of you about the research roadmap that we are talking about today, kind of how we got here. Um, Charles, can you please work with Michael? He's still having trouble getting in. If you can send him some information, that would be great. Uh, so we're really excited to have this conversation with all of you because we, feel really strongly that this is a great step forward for the SCN8A community, and we're excited to be doing it together along with lots of partners and who are other patient advocacy organizations and researchers and clinicians, and of course with caregivers and families who are really at the center of everything that we do. So. This is a compilation of 9 SCN8A kids you see on the screen here. I'm sorry, 9, 11. Um, and they were featured during the roadmap posters with these kids about their stories and, what really matters most to them, what they're proud of, what their goals are. And that really was important to us to really center a lot of what we were talking about throughout the meeting. On these stories representative of the wider group. So that was really our intention, was to continue the work that we do with a really focused, caregivers and families priorities at the center of all of that. So a couple of quick things. So those of you who have joined as attendees, you don't have to worry, you're off camera, you're muted, so don't worry. I encourage you to please use the chat or the Q&A throughout. We will have some time set aside for questions, both in the middle and at the end. Our plan is really to start. Michael's going to give some overview of kind of how we got here. Then we're going to share the results of some of the surveys that we did in advance of this meeting, which were really powerful. We did a caregiver survey, we did a clinician survey, we did a scientist survey, and we did meetings with industry partners in advance to really get the sense of what people really thought were the most important pressing issues in SCN8A research to help us guide kind of where we are headed. So with that being said, I will also tell people that we have a translation tool in that we are using today, and Charles will drop the link into the chat. So if you need translations during the meeting, you can click on the link and choose your language and hopefully follow along. And with that, I'm going to hand it over to Michael. Are you there, Michael?
Yes. I finally got on.
Excellent.
Sorry about that.
No
Um,
worries.
all right, you want to share the slides?
Yeah, Charles, let's go forward. And Charles, if you can advance the slides and give Michael control, that would be great.
You have control, Michael.
So you can just use the arrows on your screen. Uh,
I don't see the slides. I just see the pictures of all the kids and
yeah,
stuff.
Yeah.
press the forward arrow and see if that does it for you.
Forward arrow.
Or do you see the arrows on the screen you can click?
Oh yeah, there they
Yeah,
are.
try that.
Control. There we go. Okay. Now that I've fallen behind, I will try and zoom through these a bit. How long do you want me to go? 5 minutes? All right. So, basically what I want to do is this is what we did exactly in Boston at the meeting. We gave these slides, and I want to just sort of run through what we did to fully apprise you of what happened in Boston, which was a wonderful, wonderful meeting and very successful. So basically, the first thing we did was we wanted to acknowledge the tremendous progress we've made since gene discovery in 2012 to 2024. All the things, these are just some of the many, many milestones along the way. We have mouse models, we have ASO studies, we have registry cohorts, we have the Alliance, we have publications, we have phase 1 trials for 2 companies. And then we had the consensus guidelines that were published this year. And all this was accelerated through your contributions as families with data to the registry and to the organizations that have formed around SCN8A to accelerate all of this. And meanwhile, the scientific community's published over 400 publications in the last 12 years on SCN8A. So as you all know, the International SCN8A Registry has been something that's near and dear to my heart, and it's really something that's made a very big contribution to our understanding. It's one of many, many pieces. And you know that we set this up in 2014 to describe the disease spectrum, to identify genotype-phenotype associations, to see if there were patient subgroups within the spectrum of the disease, then to aid in clinical standard of care and trial design. The registry can do all of those and has done. And we have published over 13 papers along the way based on your contributions to our database. And this is just a little screen view of some of the titles of the papers we've published recently. We've accelerated the rate at which we're publishing because we now have over 400 of you who filled out the registry. So let me just cover some of the big areas that the community as a whole has made progress in. So we knew there was this phenotypic spectrum pretty early on, but it's been filled in with research from many groups. Shown here at the bottom of the slide, some of the major publications that allowed to see that the spectrum runs from no epilepsy, just behavioral and movement disorder issues, to severe DEE. And you're all very familiar with all of those. Then we've also understood through all of the data that we've collected and managed to publish in 2023 that it's not just a seizure disorder. As you all know, there are many, many comorbidities. And this is one of the central topics that's come up at the meeting, and an area we want to do better in understanding the mechanisms and how to better treat these comorbidities. As you all know, one of the big questions is, what do I have a loss or gain of function variant? And this work has been contributed, many researchers have contributed to understanding whether particular variants have gain or loss of function properties or mixed gain loss function properties, which makes things more complicated. But we have understood that there are 2 very different wings of this disease, if you will, that are quite different in their presentation and in the treatment that will be required to improve quality of life. And as I mentioned before, a tremendous amount of progress is made through labs around the world working on particular variants that are recurrent and studying them both in vitro through biophysical experiments, electrophysiology, and in studying the in vivo effects of these variants in patients like you. So what we've managed to find is that there is a correlation between particular variants that you may have and the outcome or the severity of the outcome. And it correlates very well with in vitro studies. And this has been seen with multiple labs. One of the big breakthroughs, I think, is being able to identify patient subgroups within this broad spectrum, because each patient subgroup not only has a different prognosis, but also may have a different response to treatment. So work that has been done by Elena Gardella and her group in Denmark and our group here in Arizona, as well as other groups, we've been able to delineate what some of these subgroups within the spectrum that do in fact are clinically important and may have different treatment requirements. This led to the 5 phenotypic groups that were part of the first consensus on diagnosis, phenotypes, treatment, prognosis, and comorbidities that got published in Epilepsia this year. Gabi and Jayetta took the lead in making this happen logistically and put together an incredible team of clinicians around the world to get the first consensus on how to treat this disease, how to diagnose it, what kinds of outcomes there are, And those 5 phenotypic groups are part of what I was just describing that were defined by the work that we've done with the registry and the Gardella group in Denmark and others. Let's see if I need to forward this to make the mouse show up. There it is. Thank you, Madeline, for making this slide for me. So there's been a tremendous amount of other work that I'm not going to have time to cover here, but really was a central part of the meeting. And that is, what kinds of models have we developed in the laboratory that allow us to better understand the disease mechanism and how to better treat this disease? And you really need to have these mouse and animal models. We also have iPSCs, as you might be familiar with, cells that can be differentiated into different neural types. And then tremendous amount of work that's being done to understand how this disease expresses in neurons in the brain, maybe even in different body parts. And so that was another major topic of the meeting. Okay, now here's where we turn the corner. Despite all this great progress, we know as families that our lives have been improved by the community that has developed and by the research that is done and the tremendous progress, but there's still many things ahead of us that we need to think about. And the purpose of the meeting was to accelerate progress, to fill these missing pieces of the puzzle. And some of them are listed here. Disease mechanisms are not fully understood. Anti-seizure medications are inadequate. They're not selective to NaV1.6. Polypharmacy is not well studied. There's a hit or miss sort of thing that happens when we try different drugs. Quality of life and outcomes are not improving to the extent we hope for. Treatments for comorbidities are lacking. Mechanisms of non-seizure outcomes are not understood. And there are no strategies to deal with the high premature death rate. There's an incomplete understanding of the natural history of the disease. We lack biomarkers to track disease progression and treatment response, and new smart drugs and gene therapies are a ways off. So how do we accelerate progress? We take stock in having multi-stakeholder collaborations, and this is the underlying structure and purpose of the research roadmap that we're discussing today. All right, I'm going to get off in a second. I'm just going to say that the next part of the talk will be given by those who are going to report on the surveys. We had 4 surveys: a caregiver survey, clinician survey, scientist survey, and biotech survey. All of them were crafted with the idea in mind that we needed to link the different perspectives of these stakeholders, where the gaps are, where their research priorities are, and having a central role for families to give their input on that. And so we're going to hear now about these different survey results, which I feel is a major contribution of the roadmap in itself. And then we'll go on to what was the outcome of seeing the results of these surveys. All right, so that's where I'm going to stop.
Yeah, thanks, Michael. And these are just to get us started. This was the participation from across the community. We were really thrilled. Oops. It's moving on its own now, it seems. Yeah, there we go. So we were really thrilled that we got 175 caregivers from around the world who participated, over 40 clinicians and over 20 scientists, and 8, no, we don't have them on here, but 8 pharmaceutical partner interviews were held in advance, which you'll hear from Chris about. So in terms of the caregiver survey results, I'm really excited to be able to share these with you, and we'll spend some more time digging into these as a community. And these are kind of some broad results. So we were really excited that we had representation from across 6 different continents. You can see a wide variety of countries represented here, which of course is really showing the global, we know that this is not a disorder that is, stuck in, that is more represented in the United States. That's just a matter of our, the work that we do. But, in general, this is going to be as prevalent anywhere in the world, just because of the way the genetics are, of course. So we asked families, which is a higher priority for you? Is it getting control of seizures, or is it improving non-seizure outcomes? And you can see that improving seizures was slightly higher. We know from other studies that the families prioritize seizure control when seizures are bad. If you ask family, do you want to, improve seizures or non-seizure outcomes, they're going to say seizures if there have been recent seizures. So, some of this might change given the day that you ask any family. These are the priorities that came out on top. Receptive communication and expressive communication were very, very high, and this is across the DEEs. We know this from the Inchstone Project that those are really the priorities family have, or wanting to be able to better communicate with and hear from their children. You can see use of arms and legs. So some of these gross motor, hypotonia, lots of challenges for us to be looking at here. And this is what we're going to dig into more with the community and try to figure out where we get started and start working our way through these concerns. We want better anti-seizure medications, combination therapy, and off-label medication. You can see the darker the colors here, the more people that said that was the first thing that their first ranked choice was to see new, more effective anti-seizure medications with fewer side effects, but also wanting to see, use of some of these but are now off-label medications like cenobamate and fenfluramine and better understanding of the efficacy of combinations of drugs. We have anecdotal stories about that, but not a lot of data. And of course, drug repurposing. These are other treatments that families said they would like to see more research on, alternative therapies, combinations, disrupted behavior. In the last few years, the population in our community where behavior is a significant challenge has grown as our loss of function community has grown. And then, of course, some of these other interventions we want to dig into more. A little delay here. Doesn't seem to want to move forward. There we go. We also asked people if they would be willing to participate in clinical trials, and you can see that there's an overwhelming number of families that are really interested in gene therapies. I think this is something we're going to start talking a lot more about in the SCN8A community, what these are, what they look like, what the ethical issues are, what the considerations are, because some of these are, one and done, right? So you get the treatment and then whatever good or bad comes from it, you're stuck with. So lots more talking about that soon. And I think my last slide, if it'll pop up. Nope. Okay. It went into, wow. I think it caught up all at once, the 8 times I pushed it. So I'll leave it there and pass it over to you, Mark.
All right, thanks, Gabi. So I'll spend the next few minutes talking a little bit about the clinician survey results. So we had quite nice participation. We had 46 clinicians from around the world who participated in these surveys. And sorry, there's a little bit of a lag. Okay, so we wanted to get an idea of how these clinicians tended to spend their time, be it in clinical care versus research endeavors. And as you can see here, a full 3/4 of survey participants had some amount of clinical research as part of their day-to-day work. Now, the amount to which each individual clinician was participating in research varied quite a bit, as you can see here. But I think it's fair to say that, somewhere between around 25 to 50% of the majority of clinicians' efforts was spent on various clinical research projects. It was also quite an experienced group of clinicians. As you can see here, about 2/3 of folks had at least 10 years worth of experience in treating individuals with epilepsy or other central nervous system disorders. And about 1/4 more had somewhere between 6 and 10 years of experience. Now, in terms of the number of clinicians who were actively treating individuals with SCN8A related disorders, there was a bit of a spread here. And I think that sort of highlights that even a group of highly experienced clinicians can still have a variable amount of experience treating individuals with SCN8A. The most typical response and sort of the median was about 5 patients or so, with a spread that was, as you can see, quite broad. Now, in terms of which phenotypes the clinicians were most familiar with, not surprisingly, I think the majority had more experience with treating individuals with gain-of-function phenotypes, be that the severe DEE, mild to moderate DEE, and some experience with selfie. That being said, there was quite a bit of experience as well in treating children with more loss of function phenotypes like neurodevelopmental disorders with generalized epilepsies. Now, we also asked the clinicians what sort of priorities they had in terms of how to approach, or what questions were most important in terms of the diagnosis and phenotyping of SCN8A related disorders. And we had a bit of a tie at the top. The majority of folks wanted to invest some time in really characterizing in a detailed way the phenotypic spectrum, and validating and extending the key characteristics of the 5 consensus phenotypes that were arrived at during the consensus guidelines assessments. When it came to treatment priorities, clinicians sort of overwhelmingly felt that establishing some best practices for choosing first line and adjunctive anti-seizure medications was quite important, as well as more information about how to properly dose these treatments and which combinations were the most meaningful in terms of getting more benefit for seizure reduction. There was also quite a lot of interest in trying to evaluate best practices for adding or weaning of anti-seizure medications in an effort to try to minimize polypharmacy. Now, on the prognosis side, there was the most interest in trying to identify clinical features that predict outcomes in each of the phenotypic subgroups, as well as efforts to try to identify biomarkers that were informative or would be informative in really helping to understand how disease progression is occurring, how treatment response is going, and much of this, these biomarkers, we recognized were going to be important when it came to the design of clinical trials in the future. On the comorbidity front, the clinicians felt as though more research into the sleep disturbances associated with SCN8A related disorders was the biggest priority, and then movement disorders and how to better treat those were quite high up on the list, as well as how to better approach speech and communication issues. Now, in terms of the importance of research into a variety of different areas, the clinicians felt like we needed more effort directed towards treatment and therapeutics, as well as how to better diagnose and use the phenotypic subgroups to understand SCN8A. And clinicians felt as though using the SCN8A registry as it exists and collecting real-world longitudinal data offered sort of the biggest promise in terms of research methodologies to really be able to fill in some of those knowledge gaps that exist today. And clinicians also felt like attempting to move forward with a prospective natural history study for SCN8A was going to be one of the most important projects to try to accelerate progress in reaching some of those research objectives. And with that, I'll pause and sort of turn it over now.
Thanks so much, Mark. Next up, we have Madeline.
Hi, everyone. I'm going to be summarizing the results from the scientist survey that we did. I did condense this quite a bit from what was presented at the meeting, just due to the time constraint I was given. But I'm happy to answer any more questions offline about this. So we pulled about 23 laboratory scientists. You can see the number of researchers in terms of the years that they've been working in SCN8A. So some very experienced ones, and then some who only been working for the last 5 years. We also included some researchers working on SCN2A, kind of other disorders that are related. And so this is the expertise of these researchers. So lots of expertise in mouse models of epilepsy, in gene therapies, in cell models where we can grow neurons in a dish, and different ways to study them, and in the function of ion channels. And so really the questions were centered on 3 main areas. One is kind of understanding how the disease works, so the mechanisms of the disease, and how do these mutations in SCN8A actually cause all these things that, our children have. 2 was then, how do we model this? How do we study this? And so going, range of models in the lab, from cells to mice, which are the most common model used. And then the different therapeutic approaches that can be used that we want to develop. And we really tried to ask questions for both loss and gain of function, and then really to also kind of gear discussions towards both seizure and non-seizure outcomes, especially seeing the patient survey, the patient data that really emphasize a lot of importance for the non-seizure outcomes. So this is kind of a summary of kind of what scientists felt was the most important. And so again, based on these 3 categories that I talked about, with breakdown for seizures or non-seizures, or gain or loss. And so really the priority they felt was therapies for seizures, but then also really understanding the difference between gain and loss of function. Those were kind of the top 3 priorities. But it's important to contrast this with what is actually the most feasible to study in the lab, because we're limited in the lab with the models that we have, the questions we can ask. And so here you can see that the feasibility does change the order. And so it is most feasible to study mechanisms of gain of function. That's where we have a lot of the data so far. And it's pretty easy to make models with seizures, but that was kind of ranked less important because we have a lot of these already. Versus like the non-seizure therapies, which, were ranked 4th in terms of importance. In terms of feasibility, we're really the lowest because we really don't have good models for that. So it's just important to keep in mind these differences between importance and feasibilities that these scientists voted on. So in terms of mechanisms of disease, we asked them to rank the importance and kind of what was needed in order to understand the mechanism of the disease. And so here, kind of a lot of different features of the brain, what types of cells are involved, what types of regions of the brain are involved. And so, there was really kind of a focus and priority on the different cells, and really the nitty-gritty of how these mutations actually impact the protein and impact the cells in the brain. In terms of kind of what was important to develop novel therapeutics, like what do we need to understand? There was really a lot of importance placed on understanding how changes impact the whole brain. Focusing at the single cell is important, but our brain is full of cells. And so looking at that is also really important. They also talked about biomarkers, which are things that we can use to kind of measure what's happening in the brain that we can access more easily, like an EEG or blood biomarker. And then in terms of understanding the non-seizure phenotypes, they ranked highly looking at kind of behavior and cognitive issues, SUDEP, with then some of these other ones a bit lower. In terms of the models for research, They talked about kind of ranked which models are most important. And mouse models are, really important in understanding disease. But they also talked about patient-derived stem cells, which our community has, which we can make from skin cells from patients, and different 3D models that several labs are working on as well to model the brain in a dish and manipulate it more easily, with ranking other model animals as the lowest. And what can these models actually be used for? So, they do think they can be used to study, lots of like specific drugs that we can develop, repurposed drugs, but they ranked the lowest, the kind of cannabinoid and nutraceuticals. And lastly, in terms of therapies, what therapies could be the most important for these populations? So for gain of function, ASOs came in first to investigate, and then other kind of gene therapy approaches ranked a bit lower, but still quite highly. And similarly for loss of function, even though the approach for an ASO would be different, this was also ranked highest up in terms of other genetic interventions, different from drugs. But then we asked kind of what are the main scientific gaps that we need to understand to be able to get these more advanced genetic therapies that we know many of our families, really also ranked highly. And there's a number of issues that, need to be solved in terms of understanding the biology of manipulating the gene, getting gene therapies to the brain, and really understanding their effect on non-seizure outcomes. All right, and I'll stop there.
Thanks, Madeline. I'm going to hand it over to Chris Spurge now to talk us through what he learned when speaking with biotech.
Great. Hi, everyone. So these are the exact slides that I showed at the meeting. So they contain all the answers, basically, that we got from 8 biotech companies. And it's a little bit more than I can talk through. So I'll just kind of try to hit some of the highlights. So with input from the Alliance, we reached out to 8 or 9 different biotechs and got good participation. These are all the companies that participated. I'll just note that Praxis and Neurocrine were also like big sponsors of this roadmap meeting. The first question was essentially, what is your company doing? Are you in this space? They were all in 8A in some form or other. Of course, some of them developed with clinical trials, others with drugs in development. Why did they get into the disease? Their expertise in the CNS or ion channels or screening was obviously important, but also they recognized the unmet need, the well-organized community, strong prior research where there's really clear therapeutic goal for gain or loss of function, for example. But some of the challenges with developing drugs in this area are obviously that it's rare, it makes it harder to enroll for clinical trials. There's already a crowded space of anti-seizure drugs. Most patients are on, a few different drugs, and that makes it harder to design clinical trials. So we asked them about, what potential therapeutic approaches do you see as having most potential for gain of function, loss of function, and others. There was more or less the expected answers in terms of inhibiting channel activity and so forth. There was generally high enthusiasm for ASOs and gene therapy, kind of ASOs in the shorter run over the next few years, and maybe CRISPR or other genetic therapies in the longer run. Some raised concerns about potential for excessive knockdown of expression. Obviously, it's 8A is a Goldilocks gene. If you knock it down too far, that can convert a gain of function phenotype into loss of function. There was also some concern about delivery to all the relevant brain regions and neurons. Classic gene therapy like CRISPR, changing the DNA, was considered to have great long-term potential for the disease, but to not be ready today. And of course, the permanence of changing the DNA has associated risks. There was also some challenges about classifying patients one way or the other, which are important in this area. In terms of what basic research questions the biotech companies would like to see addressed in the field, defining the biology of different variants was ranked very highly by several. Understanding more about the pathophysiology of loss of function in particular, and learning what range of NAV 1.6 levels are compatible with health of the brain, which is really not well understood. And I'll just kind of skip over the other ones. Certainly, they'd like to see development of more animal models for different classes of variants, particularly loss and multifunctional ones, I think. And a natural history study was mentioned by several. And need for biomarkers. So to know if you're inhibiting 8A, can you measure to what extent your drug or ASO is engaged with that target? In terms of patient advocacy, we asked about what strengths do you see in the 8A community, and pretty much everyone said it was well organized, collaborative, accessible. They recognize there's good patient education and clinical trial readiness, having already have a couple of clinical trials ongoing. Good people and enthusiasm, and have promoted the science was also recognized, and of course the international presence of the alliance was a plus, as well as the Cube syndrome also has contributed in many of these areas. In terms of areas for potential improvement, several expressed that the community could be more united, that the different groups involved could carve out clearer niches and roles, and they suggested that we consider partnering with related DE foundations to boost the scale. In terms of what kinds of data they would like to see, many of the companies would like to see a natural history study. They also mentioned that participation in the natural history study should not preclude also being involved in a clinical trial, ideally. They don't want to reduce the set of patients available for clinical trials. And more hard data on non-seizure phenotypes so that they could potentially include some of those in a clinical trial outcome. They would also like to see a community resource for iPSCs, perhaps managed by a company or something like that, rather than dealing with individual universities would be nice. Collaboration opportunities, they encouraged the 8A community to collaborate more with the SCN8A community and related channelopathies, to figure out which endpoints are measurable in a clinical trial. And they mentioned a few other rare disease advocacy groups who have done a really good job that we could potentially learn from, including Angelman syndrome, Cystic Fibrosis Foundation, TSC Alliance, Friedreich's ataxia, and so forth. And finally, one of them I thought said something kind of interesting. It didn't really fit in the questions, but that they found that the patient and families are not very risk averse, but companies are. So that's just something, food for thought. So that's it. Pass it on to the next speaker.
Thanks so much, Chris. It was excellent and really valuable information for us to have in preparation for the meeting. All of these survey data really contributed to us being able to, hit the ground running and have some really productive conversations with the participants. I'm going to quickly answer one question that was in the chat, and I'd like others to help too, but saying what is being done to communicate the findings of the spectrum with the genetic testing labs who interpret the testing. I feel that the VUS results would change and we'd be finding more patients in the milder side of loss of function. And you're absolutely right. We are working with a wide range of genetic testing companies to discuss with them the results of this work, so that they have a better sense of the and doing more communication with clinicians as well, so that they recognize this spectrum. So the VUS results, it's a little bit complicated. Geneticist Michael, do you want to take, that VUSs are why they're called VUSs?
Well, they're variants of uncertain significance, and that there's a certain procedure that the genetic testing companies use when they find a variant in a particular patient, and they cross-check it what's known with other patients with the same variant, and that's one of the highest levels of support for a variant being pathogenic. There are gray areas, and they become variants of uncertain significance. The companies themselves are responsible for updating themselves on the literature, so that they themselves can do a better call on whether one of 5 categories of pathogenic, likely pathogenic, VUS, et cetera, down to benign is the most appropriate call.
Great. Thanks. We're going to keep pushing along, but I encourage you to continue to put your questions in the Q&A or in the chat if you have a clarification or just want to say something you're thinking that might be worthwhile for others to hear. Please go ahead and do that. All right, so we will move on because we're always behind. That's how it works. So we're going to start with Michael, who's going to give a short intro, and then we're going to hear about what happened in the work groups during the roadmap meeting. So Charles, if you can just share those slides again. Thank you.
One moment, thank you.
All right, Michael, feel free to jump in when you're ready. So
I'm looking for that little arrow.
yeah, Charles is trying to find, I think, where we left off.
All right, well, I'll just start talking and then you guys can catch up. So the next major portion of the meeting, once we went through all those surveys and we saw how things were related to one another, where the common gaps were, where the common priorities were, then we decided that the next step was to formulate working groups that could break out at the meeting. So these could be interdisciplinary research, laboratory scientists, clinicians, caregivers, biotech, breaking into these 5 different working groups that we thought were most relevant to the findings of the surveys. So I'm going to jump through in order to save. Well, so that's kind of what I'm showing here. So we have the surveys we just heard the results of. Then we kind of looked at across the surveys to find common priorities, common gaps. And from that, we found these 5 major themes that we thought were most important. And so this part of the talk will illustrate those 5 working groups. I'm going to skip through these just to give a couple of examples. So for example, if you compare what the caregivers' priorities were for quality of life versus, let's say, the clinicians. In this case, as Mark mentioned, the sleep disturbances were high up for clinicians, but they weren't, interestingly, so high up for caregivers. But we know they affect many, many families, sleep disturbances. The speech and communications was very high here and not quite so much for the clinicians. So we did a lot of integration across surveys. We compared what the clinicians and the scientists were interested in, where their gaps were. The surveys were designed to be able to do this. And then some of the shared and complementary views among the stakeholders here came out, like with seizure control. And then you can see, I'm not going to go through all of these, but gene therapy, non-seizure outcomes, biomarker discovery, registry development, disease mechanism, improving diagnosis and phenotyping, et cetera. So these are the themes that kept coming out through the discussions and through the surveys. And so we decided going into the meeting that we were going to have these 5 working groups. And then we voted at the meeting if all the stakeholders in the room agreed with these 5 working groups. And amazingly, everyone thought these were the most important areas to break out into and to push forward. So the next part of this will be that we're going to have, and you can see here the names of the working groups and then the leaders of the working groups, not necessarily everyone that participated, because each of the working groups probably had about 8 to 10 participants. So I'm going to turn it over now to the next. The purpose of the working group was to define the knowledge gaps, design a research strategy to fill those gaps, and make sure your strategy facilitated improvement in patient outcomes and addressed caregiver priorities. So that's my quick turnaround for you.
Thanks, Michael. All right. Okay. Sorry, the slides are moving slowly here.
Since we didn't have a biomarkers representative that was able to join today, I did make a couple slides, but we could choose to skip it if there's not enough time.
Yeah, I think you can just take 1 minute to maybe say why these are.
So everyone's heard a lot about biomarkers already in the discussion. They can be molecular, histologic, radiographic, physiological. They're used to assess the susceptibility to a disease, the safety risk, the diagnosis, monitor the course of the disease, prognosis of the disease, and predict response to treatment. They can contribute tremendously to the development of therapeutics, and they can be used as surrogate endpoints to substitute for clinical endpoints in a trial. So if a biomarker is responding, but we don't quite see a change in, let's say, a patient phenotype, whether it be a seizure or non-seizure outcome, that can be an indicative of the drugs doing or the new therapies doing something in the right direction before we actually have definitive proof that it's working. So these were the gaps that were identified, and then the research priorities were basically discovery, validation, and developing a standard biomarker panel for clinical use. I won't go into the details, but these slides will be made available for people to see.
Yeah, and this is where we're looking for more expertise in this to help us start to move this work forward, because it's a very specific kind of work to do. So. And we're partnering also, with other groups, like Chris mentioned that biotech said, hey, you should really partner more with 2A. Well, we have a very close relationship with Families SCN2A in the US, and we're looking at trying to collaborate on a number of things. And we just won a big CZI grant. And SCN2A Germany and Italy are partners in that. So we'll be collaborating with them as well. So I think there's lots of opportunities to see that work move forward. So, all right, gonna hand it back. Let's see.
Madeline.
Right. Yep.
Okay.
Sorry.
All right. So the group I was in charge of kind of leading with Wenxi Yu, and we had, a group of participants here, was on transformative therapies. And so what are transformative therapies? These we felt were described as therapies that could not just tackle seizures, but also improve all the other non-seizure phenotypes that we know SCN8A patients have, right? And all the work that has been done, both gain and loss of function, there's all these other things associated with SCN8A mutations. And so we want therapies that are not just, impacting the seizures, but all these other things as well. And the group members agreed that these therapies could be drugs, like small molecules, or they could be therapies that are genetic therapies. And so, so where we're at is that, we are really lucky in our community that thanks to all the research that has been done by so many of the basic researchers who are at this meeting for the last, 30 years since the gene was discovered initially by Miriam Meisler, has led to the fact that we have 2 drugs in clinical trial, drug from Praxis and the drug from Neurocrine, which is really, really exciting, and that really specifically target the channel, right? These are drugs that target NaV1.6, not lots of other things like most of the drugs that our kids are on. And these are for gain of function patients. There is no ongoing drug development on the loss of function side. And then on the genetic therapy side, where we're at is that there's been some really nice research, really pioneered by Miriam Meisler's lab, showing that different genetic approaches, that in a gain of function model, if, gain of function, which makes your protein more active, which, usually leads to seizures, if you can tone down, reduce the levels of your protein, so kind of bring it back down to baseline, you can prevent seizures in mice and also extend the survival of these mice. So this is really exciting that we have these therapies that really, again, specifically target, NaV1.6, the protein made from SCN8A, and extend survival in mice. But these are all experiments in mice. And in loss of function, there are currently no, at least published work, again, of genetic strategies that have shown any effects. And so, again, we felt that both of these approaches, could be transformative by really targeting specifically just the protein where we have the mutation, that this, could be transformative in that it affects kind of everything that is related to the disorder. So this is exciting, but obviously we need more. And so there are several kind of gaps, and I will, there's a lot more to these, so it's really hard to keep this down to 5 minutes and to provide you all with the appropriate background to really explain this. So again, I'm happy to answer any more questions offline after this. But there's a big question, as kind of is brought up, of SCN8A being this Goldilocks gene, where it's if you have too much, it's not good. If you have too little, it's not good. And so when we're trying to make therapies that change how much is present, it becomes really risky to change the levels like this, because we could, really cause damage if we go too far in one direction or the other. And so there's really a lot of science that needs to be done to really understand kind of how much can we change the levels. What's the window that is good enough that we don't go too far in one direction or the other? And these are questions that are, important for both therapies for loss and for gain of function patients. So there's a lot of kind of basic research that needs to be done. And without this, biotech companies will not really move forward with these kind of more genetic therapies. The other question is, okay, if we, target SCN8A in the brain, right? If we gear our therapy to the brain, does that help with all the kind of non-seizure effects that our children have, right? Or do we need to go after those individually as well? So does one therapy that might fix something in the brain, does that work everywhere? And we really don't know the question to that. And the problem is generally that most of our mice don't actually have many non-seizure phenotypes. Most of the mouse models that are published, and there are currently some new ones in development, mostly have seizures but don't have a lot of the other stuff. Or maybe we haven't looked hard enough to see if they do have a lot of the other stuff. So that's something that we need to do as well. Then is, when can we give these treatments? Is there a time frame where it might be too late to do that, right? When can you correct kind of or fix the brain? Can we give this therapy, at a child who's 10, 15 years old? And can that still have an effect? We don't know. There's a lot of questions about the brain generally that we just don't know. And lastly, is there a specific region of the brain that's more important? And we don't know that. So the next slide, I just had kind of a bunch of experimental plans that we've kind of priorities that we've laid out to try and address some of these general questions. And I'm already over time, so I'll stop.
Thanks so much, Madeline. I'm going to pass it over to John Schreiber, who's going to talk about non-seizure challenges.
Thank you. And Ann Berg was there at the meeting. I had a family emergency, but anyone who wants to chime in with additions that I might miss, please do let me know. This is, I think I'm looking at a slide that says development and delivery.
Yeah, you should be able to use the arrows.
Okay.
Charles
Great.
has given you the right.
Gotcha. Okay. Now I have the power. Okay. We were tasked with discussing non-seizure outcomes, recognizing that non-seizure morbidities are a significant concern across all SCN8A phenotypes, particularly those with DEE, since there is a wide range of symptoms affecting multiple systems, and this often includes the engagement of all sorts of different specialists. That's something I think we underappreciate. We're sort of overrepresented in this community by neurologists, but I think this is where it becomes important to understand that there are other experts on disease from the clinical side that we need to engage, and then start thinking about how we can address these non-seizure outcomes from the basic science research perspective. And these challenges can be really concerning to families, especially if seizures. The hope is that we can get better control of seizures. We might see better developmental outcomes, but that doesn't always work, and may not always translate, as Madeline was saying, to improvements in other systems, right? If you're targeting something that's affecting the brain, we don't know how much that's going to target some of these other symptoms that may be very important in quality of life. A recent study that some data and colleagues have looking at different kinds of DEEs show the importance of that impact of seizures relative to other symptoms. That importance of seizures decrease since the time of last seizure as that increases. Not surprisingly, but it really is important to keep these things in mind. And we know from the FDA and others that we really need to consider these other things besides seizures that are impactful to families and patients as we're thinking about developing therapies and designing clinical trials. So this came from the survey, wide range of symptoms representing various non-seizure outcomes. Most commonly things like communication. We know communication is a big thing across various DEEs, certainly very important for SCN8A. But other things too. We talked about movement disorders. We talked about low tone, motor problems, orthopedic issues, respiratory issues, GI issues. These are things that, those of us that know patients and talk to families, or, our families experience all the time. And there are things that are important to address. So very helpful to get all this data. We have many gaps. So while we have some basic understanding of what these problems are, and it's increasingly recognized that these do represent significant, important challenges for families, we still don't have a great understanding as to why a lot of them are so prevalent, right? How do they originate? What's the pathophysiology, and how do we treat them, right? Is this autonomic dysfunction that's leading to GI problems and constipation? We know that SCN8A is not just expressed in the brain. So we need to align clinicians and researchers on these priorities, so we have more attention to recognizing, treating, and then studying these challenges. I think one more slide. Whoops. And our strategy, both long and short term, is to take on research that will take into account all these different kinds of conditions, improve quality of life, and ultimately have more effective treatments that address these. It may be some existing therapies out there, right? Things that we need to focus more on, things that may not be as impactful, that you're spending a lot of time doing this or that therapy and maybe not seeing much improvement there. So we need to have a better understanding of how these different therapies, some things in existence and things that may not yet exist, how they might impact a patient's life. So again, it comes back to understanding natural history, looking not only at SCN8A, but other DEEs as well, and learning from each other, and how we can evaluate non-seizure outcomes. We have appropriate outcome measures. We know that the Vineland is not always the right outcome measure. Quite commonly, it's not, and we may want to measure inch stones instead of milestones, which Gabby and others can certainly talk more about. We want to really leverage our existing data, from the SCN8A registry that Michael and team have built, from the Danish Epilepsy Center, from other centers. I know Dr. Fitzgerald at CHOP, they have a big cohort of kids they follow with SCN8A as well. How do we use data that's in the electronic medical record and really better integrate this? I think that's something we want to better understand. I'm not seeing the bottom of the slide, but there's another important piece there that I'm probably missing. But I will leave it there. Certainly plenty of work to do in this. And sorry, the last thing I think was then being able to take this to the bench as well, and understanding disease mechanisms, and how do we develop effective animal models, not just looking at seizures, but animal models that look at respiratory problems, GI problems, all these things that we can then test with the various therapies and see what effect that has on this animal model.
Great. Thanks so much, John. That was great. Covered a lot of ground there, and there's lots of work to do, obviously. All right. So this is Mark Fitzgerald.
Yes. So Josh Hack and I co-led a group at the roadmap meeting that was really tasked with thinking about what can we do with what we have right now, right? I think we all acknowledge that some of the transformative therapies that we really hold a lot of hope for in the future are still some time away, right? Potentially years if we're talking about things like gene-based therapies. But of course, families and individuals with SCN8A need some relief now, right? We recognize that, seizures are quite significant for a large majority of individuals, and there's a tremendous number of comorbidities that also really have a negative impact in quality of life. And one thing that we learned from the consensus guidelines process is that the studies to date really focus quite a bit on single anti-seizure medications or treatments, and how those individual treatments tend to help with seizure control, right? So we learned things like sodium channel blockers for individuals with gain-of-function variants are quite important, and that's how we came to recommend oxcarbazepine or carbamazepine as first-line therapies. Or For individuals with loss of function variants, valproic acid, ethosuximide, lamotrigine, those can be quite effective for seizure treatments. But the literature really does not have anything to speak of when it comes to combination therapies. So if carbamazepine, for example, does not help with seizure control, what do you add next? What synergizes with that particular treatment? What has the potential to, what combinations have the potential to improve seizure treatments and outcomes? And we, like I said, we really know essentially nothing about that. So we got speaking quite a bit about what sort of system would we need to start answering those sorts of questions. Um, and, we spoke quite a bit about the tremendous data source that we have in the SCN8A registry. But the sorts of things that the registry is good at is collecting a wide wealth of information, but the registry tends to be lacking when it comes to certain real world data sources, like what dose of medication or what medication combinations were used at any given point in an individual's treatment journey, and how did that specific dose or combination of medication impact seizure frequency. So, what I think it's really important to recognize is that real world data can be very important for studying outcomes. And what do I mean by real world data? I mean things like registries, I mean things like electronic medical records, and other sources of data, including patient reported outcome data. Um, but all of these individual sources need to really be married in a way that can be useful in answering some of these questions. So we talked a bit about what sort of information is contained within an electronic medical record at your physician's office, compared with what sort of data is actually contained already within the SCN8A registry. And we spoke quite a bit about how very granular information about medication dosage, combination, prescription patterns, those are all really contained nicely within the electronic medical record. And there's also office visit data like seizure frequency or the presence or absence or changes of the severity of a variety of different comorbid conditions. Part of the problem with electronic medical record data, though, is that it's not always standardized. The prescription data is, but some of the outcome data is not necessarily recorded in a similar way from patient to patient, from institution to institution. So it can be challenging to get good information on combination therapies and their impacts solely from an electronic medical record. On the other hand, the SCN8A registry contains very good standardized caregiver entered data. And much that we have there about seizure frequency and comorbid conditions is much better than what exists in the average electronic medical record. What tends to be lacking and has really impeded some of Josh's work on machine learning approaches to try to understand um, uh, treatment implications of anti-seizure medications and their doses is that the registry really doesn't have granular enough information about the medications, about what changed at what point in time, and what was going on with, say, seizure frequency at the moment that those changes were made. So what we discussed really is a solution and what we think is necessary to make more progress on this particular topic is really more granular data, but specifically on what is going on from a seizure frequency standpoint on a daily or weekly basis, right? The registry has certain individual time points. Your clinical notes in your electronic medical record have individual time points, perhaps 2 times or 3 times a year. But to really gauge what's happening in between office visits or in between registry data entries, we need more information in those gaps. Because as you might imagine, if there are multiple treatment changes being made between office visits, it's difficult to know what impact any individual change has had on seizures, because by the time you get to your next data point generated either by the registry or by your clinical notes, quite a lot of time has passed, and you're really trying to reconstruct things in a retrospective way that doesn't work terribly well from a data analytics standpoint. So the solution we really came up with and we talked about quite a bit was how can we marry these individual data sources and create one data set that has all of this important granular information contained within. So really the solution we thought of was a a combination approach where we take what the EMR is good at, particularly the medication data and what changes happened when and in what order, extract that information, combine it with the existing data within the SCN8A registry, and also combine it with a new real-world data source, which would be caregiver-entered seizure frequency data on a daily or a more realistically weekly basis. And then once that data set is constructed, then we can start to analyze trends in what's happening with seizures as a function of individual changes with medication. Because in this combined data set, we would actually have all of that important information in one place in a usable fashion. And then once that data set is there, then different machine learning approaches can also be utilized to try to understand this data and make predictions about what medication combinations or what dosage combinations or drug levels might actually be more likely to lead to a meaningful seizure reduction compared with other combinations. And I know Josh is on the call now, so if he has anything to add to that, I'd sort of welcome him here. Otherwise, I'll sort of turn it over to our next speaker.
Yeah, nothing at the moment. Thanks, Mark.
Thanks to both of you for working on this and moving it forward. It's already making some progress, so thanks for that. All right. Charles, can you move forward to the next slide? I think we we're not going to do biomarker right now, but we're going to go and move on to Jeremy, if you're here.
Yep.
Great to have you, Jeremy. Thanks for taking the time to present today on behalf of your group.
Yeah, thank you everybody for joining. And I'm kind of the new person into the team, and this is such an amazing society and group, and I applaud all the work and everything. So I think you'll see the whole brain, whole body working group. I think you've heard a lot of the trends and themes that we've had to this point, and I don't know if I have control over slides.
Yeah, you should be able to move it either with the arrows on the screen or your
One second, Jeremy. You do now have control.
There we go. All right, awesome. All right, so I think you'll see a lot of the themes that we talked about throughout all the other working groups really reverberated through the whole brain, whole body working group as well. So some of the key gaps that I think we've already heard several of these. What regions of the brain is SCN8A variants impact? How do different variants impact different cell types of the brain, especially thinking about gain versus loss of function? How do the variants impact what we call the connectome? And there have been several big news stories over the last actually week over these establishments of these what we call connectomes. So how do all the neurons in the brain connect and drive tracks and activation? And so asking these questions about how does SCN8A really impact those networks of cells? What other factors contribute to these different responses in treatments that we've heard? So there are other genes, is there pharmacogenomics that we need to be accounting for that would be useful coming from whole brain, whole body dynamic mapping? And what regions of the brain outside of the brain cell types contribute to the diverse phenotypes that we've heard about? And are there any other genes and variants outside of SCN8A biology that are impacting some of this? So are some of the families that have same variants that have subtleties and differences? What else contributes to those differences? So some of the research priorities, we kind of set some of those that are more immediate versus those that might take a long time as we start setting these priorities. One of the things that came up, and I may have been the person that helped drive a lot of this as I've joined the group and I'm a big data lab, how do we bring all the existing data sets? So I think it was mentioned CZI, where the founder of Facebook has invested huge quantities of money into understanding all the cells of human. And those resources can be mined to understand SCN8A. Founders of Microsoft have invested huge resources in brain mapping. So bringing that data together, and we're already starting to work on and structure a paper that several people are coming together. If you have interests in helping contribute to that as a researcher or a scientist, please feel free to reach out. Also, I mean, into those brain cell maps and regions outside, bringing those landscapes together with additional insights of other sodium channels. So is there some redundancy and biological overlap in certain cell types? And do they contribute to certain neural tracts that's not just, is SCN8A expressed? Do we get impact in those cells in a loss of gain of function, especially loss of function variants? Enhancing some of the patient medical imaging integrations, animal model imaging integrations that exist, and then enhancing some of the diverse phenotypic discoveries that have come up, especially around some of those variants of uncertain significance, where some of those phenotypes might not even be recognized yet because they do not get diagnosed as likely pathogenic or pathogenic. Wait for the slide to advance here. So some of the intermediate projects, I think we've heard about the different brain regions and the circuits. Can we use our mouse model systems to enhance those? Can we also take our mouse models and get additional resources of different variants into the mouse models, especially covering more loss of function in those? We've heard about some of the stem cells and making mini brains. Out of those, can we grow the number of variants that we have within those systems as well, adding loss of function in them, looking at different regions, different networks. And then one of the areas that's kind of intermediate to actually more rapid is, can we identify experts outside the field of SCN8A that might help support some of the diverse phenotypes? So bringing in sleep experts, bringing in GI experts to really gain more insights there as well. And then some of the long-term goals, I think as we have heard so much already, is the non-seizure epilepsy phenotypes. How do we increase the studies of those using iPSC models, using mouse models, and really start setting those alignments to having the tools, especially to think about the genetics and the therapies around those. There was some conversation about establishing co-modifier mapping programs. So are there other genes, as we've heard about the Goldilocks concept already, are there other genes that we could also target that might be a little bit simpler of a Goldilocks dynamic to think about in some of this? And then I think the biggest theme, I think, throughout is, how do we scale all of this data modeling using AI and machine learning strategies to integrate all of the resources we have from human phenotypes into mouse models, into stem cells, into the diverse genetics, and really thinking about that bigger integration point. And that's, I think, I'll pass it over then.
Excellent. Thank you so much, Jeremy, and to all of the group leaders who took the time to help us prepare and get ready for this meeting, and then have really productive conversations during the meeting. So we're going to transition, much like we did at the meeting itself, to have a panel of caregivers talking about what the meeting was like, what was most important to them, what they felt was meaningful. And I'm just bringing up all the families now. who are going to participate, and we'll hear from them about their experience there. So taking 1 second here to find everybody. And there we go. All right, I think that's everyone who's going to participate. If you're not on and you wanted to participate, just ping me. But I think these were everyone who's going to chat about the meeting itself. So we've got Casey on, who's the executive director of the Acute Syndrome Foundation, who joined us. We're happy to have you with us. And Joanne Kelly, who is a leader with SCN8A Australia. And then we have Tammy and Megan and Mary, who are SCN8A family ambassadors and do a lot of talking with families and connecting with families and counseling them. So we also had a number of other families who were with us as well, including representatives from SCN8A Spain and SCN8A Italy and a couple of others. So, I just wanted to give an opportunity here for you all to talk about what was really meaningful for you at the meeting, what you are inspired to see more research around, just get some reactions from you. So whoever wants to jump in first, whoever unmutes first gets to go.
I'm unmuted, so I guess that's me. I guess what struck me was just the fact that the research has really been propelled by parents and caregivers who are researchers or scientists, and just that level of enthusiasm that has really propelled the research forward so quickly. So of course we're so grateful to all of you for doing that. Also for me, just meeting in person the other families, that was really powerful for me, and something that I have probably underestimated. My daughter's now 18. So in Australia, we don't have a lot of families identified as yet. So having the opportunity to go to the States and to meet the other caregivers and all the doctors and researchers that I've only seen on a screen or in, on paper, that was really amazing for me.
Great.
I can
Casey.
kind of piggyback on
Yeah.
that. Yeah. So I just wanted to kind of mention, when you sit down in a room and there's, how many people were there? At least 40, right? About
A little
40,
over 40, yeah.
40, 50. And I'm looking around the room and I'm like, dang, these are like the SCN8A epilepsy rock stars, right? I kind of felt like I was in a room full of celebrities. And so I just think that speaks very highly to our community, and how, even though it's small, we have these amazing people that are behind us and really wanting to move mountains. And just comment on the energy of the room, the energy that everybody was there for a reason. Everyone was very invested, very involved. And the amount of, that everyone is extremely eager and willing to hear about families' perspectives. Never once did I feel like my opinion didn't matter. I felt like it was actually sought out, and was an important, kind of driver in where everything goes from here.
Yeah. Yeah. Thanks for sharing that. I definitely was palpable in there. Yeah. And lots of rock stars for sure. Not all of them, but lots of them. Anyone else? Tammy, Mary, Megan, anything you want to share? Go ahead, Tammy.
I think that the two girls covered most of it, but it was the collaboration, the enthusiasm that everybody shared. Like, everybody was there for us. They were there for our kids, for SCN8A. And, to me, it was the most amazing thing I've ever been a part of, mainly because the families, I mean, I love everybody, and the researchers, they were just amazing. All the scientists and researchers, and if I missed anybody, but everybody was amazing. And everybody was there for us. Like who's there for just a kid that was like within the 150 kids discovered with this rare disease? Like, all these people care. They care about our kids, and even the adults with SCN8A. And I just feel like this was amazing. And I was proud to be part of it.
Thanks, Tammy. Mary, I see you're unmuted.
I just felt validated. I felt like it was such an amazing well-rounded group of people. And when do you ever have the opportunity to meet in a room with so many experts, some from so many different areas? You know, typically you're just meeting with a room full of doctors or just the scientists to get together. And this was everyone collaborating, everyone who had a vested interest in moving research forward. And that's just so exciting to me that everyone collaborated and was there for a common purpose.
Mm. Megan.
I was very humble to be in that room and see everyone having the common goals and everyone was respected. Um, and even when we left and we were all back at the hotel, so many people are down in the lobby and outside till 1, 2, 3 o'clock in the morning, and we're back at it at 7, 8 o'clock in the morning. Um, it was amazing to see. And I was in a group with Mark and Josh when we did the breakout sessions, and they really listened to Kristen and I because we were the caregivers in that group, and saying what the information they needed and how do we get there, and us being able to vocalize and also say our caregivers are also overwhelmed with a day-to-day life, and what is the bare minimum that you need to reach that goal. And for them to truly listen and feel respected, and they respected that our community is overwhelmed daily.
Yeah. And they're moving forward with it, which is really exciting.
Yes, yes.
With it. Yeah.
I was just also going to mention too that I appreciated the looking at short-term and long-term goals. Obviously gene therapy was a big thing that our families were wanting, but that's not today. So let's look at what's today, but also be looking at the long-term goals and what could be tomorrow. So I appreciated the both aspects that we looked at during that meeting.
Yeah, and I think that's going to be really important as we're prioritizing, thinking about what can we do now, right? That's going to help quality of life. And then, the cure is obviously that's our ultimate goal. And there's lots of steps along the way that we can take as a community to try to, incrementally improve the lives of our families. So it is really exciting. And one of the other things I wanted to address with you all is, what came up at the meeting. We had all these surveys, and it was really interesting to see where the family, the caregiver priorities were, and the researchers and the scientists, and then having all of these work groups talking and moving, and you could see what some of the ideas that came up. I mean, is there something that you're particularly excited about moving forward, seeing more research on, or involvement in? And not everybody has to answer. I'm just curious if people have.
I guess I can start off. I know even today, there was a lot of discussion about seizure outcomes, but we did have long discussions as well about non-seizure outcomes, behaviors, GI, pulmonary issues, things that impact daily lives and have also been life-threatening to children. And I'm looking forward to the collaboration and, exploring the commonalities between SCN2A, and possibly SCN1A, and banding together and really addressing some of these other non-seizure outcomes that our children suffer with.
Yeah, and I think we're going to be really cognizant about making sure that at any given time we're prioritizing not just, one for one group, right? We were thinking about how do we really prioritize for gain of function and loss of function and different kinds of challenges along the way.
I was just going to mention too, talking about the possible IPSC repository, because I know our families are all willing to help in any way. And so I think if we can figure out what's the best way to collect that data, and I really think that our families will come forward and give whatever they need to give. So I think that was excited to think about that and have a collective place that we can put that. And then also, I appreciated us thinking about bringing in the specialty people of like sleep, GI. I think so much. Sometimes we get stuck in our 8A silo, but in some of these things, I think we absolutely can branch out into some of the other specialists. Can't speak tonight. So that was exciting to see and to look forward to.
Yeah, I'm glad you brought both of those points up, really important ones. And I want to share also that I think, we're a community that is small, right? We don't have a ton of people. And so a lot of what we're also thinking about, and that came out of the meeting, is thinking about how do we work collaboratively. And I think that especially on things like sleep and behavior and those kinds of things, there's other communities that have thousands of kids that are dealing with the same issues. And it's not likely that it's the etiology of that issue is that specific mutation. There is some stuff going on in the brain. How do we learn more about that? And really collaborating to do that. So we have larger numbers, we have a pot of money working together. So, we really would love to drive and move some of that forward as well so we can learn more.
Okay, I'll go next. So I think I might have been in a really complicated group. A lot of it was way over my head, but one thing I took away from it was the talk of using the information that we already have. Like we have this information in a data pool somewhere. Let's like run, analysis on it and let's use what we already have now and get information. Jeremy can explain all this better probably, but we have all kinds of information everywhere and we just need to use it. But how do we use it, in the best ways?
Mm-hmm. Yeah, and I think there's a lot of potential there about how we can, we have big data, and I think we'll start, bringing in some big data people. How do we do this? And, Hammer Lab is already doing some of that, right? Using it to do predictive modeling and, working on predictions around best medications. And, there's really some cool stuff happening there. But,
Yeah,
and
Casey.
Citizen and, right.
Casey covered the other part I was talking about, the other testing that we can do that is less, invasive with the skin cells and stuff like that. I mean, things like that we can start with, instead of doing the whole, genetic gene therapy,
Yeah.
We got to work on that first maybe and
Yeah.
move forward.
Yeah, I just want to acknowledge that we're over time. So I think we are going to take a few extra minutes. I'm going to take moderator's privilege. And if anybody needs to hop off, thanks for joining us. But I think a couple more minutes here and then Q&A. So if you have questions, feel free to drop them in the Q&A or in the chat, and we'll get to them in just a minute. If anybody else here wants to answer to what you're excited about, or if the meeting, helped adjust any of your expectations about how things with science and research works. We all want it to go faster, but that's not always so easy. And if you have thoughts on how, researchers can improve their communication with caregivers, I think these are silos that we're all working to try to break down. So I'll leave it there, and anyone who wants to have some final comments on this panel about that.
I guess going to the labs and seeing how the research is done in different universities and how reliant they are on funding. So the importance of the pharmaceutical companies and that funding is really key. So I guess for me as a parent, I didn't quite understand the importance of that. So it's really it's essential. Without funding, the researchers don't have grants to go forward and do this work. So that was an eye-opener for me. So yes, we'd all love it to go faster, but it's really dependent on funding.
Yeah, absolutely.
It seems common sense, but yeah, it kind of lowered my expectation a little of how fast things can go or where the money comes from.
Right. Mary, Megan, any final thoughts?
I think it was important that we were able to speak as a panel, as caregivers, and I hope that the doctors in the audience heard some of the complaints and concerns that we all hear from parents. One area that's a huge area of concern is how many doctors in the country and internationally have never heard of this, and getting the information out to those people so they know, who's an expert that I can reach out to. How should I be treating this? What else do I need to look for? What medicine is appropriate? That's huge to me, to be able to contact as many physicians as possible to let them know about this and the research and get them excited about it as well. Because for the loss of function families, you may never see a neurologist. If you're not having seizures, you may have no idea that you even have this disease. So to spread the word about SCN8A, especially the loss of function area, I think could really be very beneficial to so many families out there. And I think it would really broaden our numbers, to see how many people actually do, in fact, have this that we don't know about. But I think we need to rely on the doctors to spread the word about that. We can't do that, but I hope they got that message from us.
Yeah, it's really important. All right. Any last word from you all? I want to thank all of you for being at the meeting, for representing our community, the voices of the entire community. They all obviously couldn't be there, but it was really important that the meeting was centered on caregiver and patient priorities. And I think your voices at the table were really critical. So thank you all so much, and for helping keep the meeting running. You guys were essential to all of that as well. All right.
Thanks, Judy. I might just say one more thing.
Yes.
Just with sharing information to doctors, because it's not very well known in Australia. There are a couple of experts here, yes, but even my neurologist, who was the head of department at a very big hospital, I shared the latest consensus with him, and he said that was very, very useful. And he would pass that through his to all his contacts. So we can play, I think, as parents and caregivers, we can play a part in disseminating that information. So just making sure that our own doctors are aware, and then that they can then pass that on to their contacts. I think even if we let one or two more doctors know, then that can help spread knowledge.
Yeah. Great point.
Well, like Michael just said, how can the researchers do to improve communication with us caregivers? A lot of the times I have no idea what's going on, or if I'm needed for a study, I'm willing to participate in most everything. So, if you get a study where you need a skin sample or a blood sample, how do we, participate in that stuff, and, what can we do to help? Because I'm more than willing to do most everything that I might, that might help my son and the other children.
Yeah. So thanks for that. All right, so I'm going to take us back and have us go back to the larger group, and talk about a little bit about if there are questions from the community. I'm going to ask people to go ahead and chime in. We've got one that's in, let's see. We've got one that's in the Q&A, which is, coming from a positive viewpoint without promoting false hope, what do you think is best case scenario for a significant breakthrough in treatment? ASO, new anti-seizure medication, et cetera. Are we looking at 5 years, 10 years more? And thanks to all of these amazing presenters. Thanks for that. So, Madeline, do you want to take in terms of genetic therapies?
I mean, I think, as I mentioned, that, we have these 2 clinical trials with 2 new ASMs targeted against NaV1.6 that are ongoing, and there's still more to do before they can get to people, but that's really the closest that we're at in terms of a specific, potentially transformative therapies. And there was recent data being released from Praxis showing really encouraging data on this. So, that's like really exciting. ASOs are still quite a ways away, especially for a larger population, because as I mentioned, there's still a lot of basic biology that we need to understand in order to be able to use this technology for SCN8A, because of how complex this gene is. So I think for that, we're definitely looking further down the line, more like 5-10 years, along with kind of some of these other genetic therapies like CRISPR, that, or other kind of gene therapies where, there's lots of technology development that needs to happen. It's not specific to SCN8A per se, it's more the technology itself. And so there's lots of efforts related to lots of genes and lots to learn in that sense. So, we're not going to be working in isolation on this. And this is more relying on kind of the efforts of other people developing therapies like this for other genes. But definitely we're further away from that. So that's kind of, I think, where we're at.
Anyone
think
else want to weigh in? Yeah.
Yeah,
go ahead.
I just say quickly, I think we did have it built into the meeting agenda that we were going to balance our portfolio, our investment portfolio into short-term and long-term investments. And even on the short-term side, we know we've got a ways to go. But we also don't know exactly where the next breakthrough is going to come, whether it's a gene therapy, a new drug from a pharmaceutical company, or something else, transcranial magnetic stimulation. We talked about all these things at the meeting. We want to cast a broad net to make sure we're on it. But at the same time, we have to move forward with realistic investments as short-term and long-term. So these are the critical and challenging questions that we're faced with.
Yeah. I don't see any other questions, but we'll take another couple minutes if anybody wants to drop anything. Anyone else want to weigh in? Manoj, we haven't heard from you. If there's anything you want to say about your group or the meeting, or give you a chance to weigh in.
Sorry, I was on mute. No, I think Madeline is correct. I think with the pharmaceutical companies that are invested in SCN8A, I think those clinical trials are going on right now. I think we'll hear back in the next year or two, and gene therapies are moving very, very rapidly, particularly in other diseases like spinal muscular atrophy. So I think it's a waiting game in a way, but I think we're getting there, and I think 5 years we'll have something.
Yeah.
And it seems like that was like one of the main points that everyone was talking about is new therapies, because that's kind of the most important area, right? We want to try and treat these seizures while we continue to figure out the mechanisms for these seizures in animal models.
Yeah. Great. Any other final thoughts from our esteemed panel here? Terrific. Let's see. So we've got a comment here. As discussed here, a very feasible approach for SCN8A research is focusing on behavioral assessments with higher resolution, which could provide valuable insights into clinical trials and various interventions, both pharmacological and genetic. The more we understand about the etiology of the disease, the nuances of behaviors and correlations to environment. Yeah, this is a really good point, Dylan. We still have a lot to learn when it comes to SCN8A and the connections. And earlier there was a question about, behavioral challenges. Do they have to do, with trauma, with genetics, with, what's going on there? I think there's so much still to learn. And that's the point, is we're going to keep digging and keep trying to learn together. All right. Well, thank you everyone for joining us from this panel. I'm going to, Charles, can you bring up that final slide, please? And I'm going to do a quick thank you, and then Michael is going to share some final thoughts with me about where we're headed. All right. So first of all, a huge thank you to everyone who was involved in planning this meeting. Madeline and Jayetta and Michael and I and Kelly, one of our staff, spent a ton of time getting things ready. Madeline and Chris hosted us, which was incredible. And we're really grateful to their support there for really hosting an amazing meeting and doing a lot of the legwork there. Thank you to Praxis, who held our dinner one night, and Madeline and Chris, who hosted dinner another night. And really, I think that there's so many people that were involved here that were really critical in making this happen. So Charles, if you have that last slide, that would be great. So we want to take a minute, of course, to thank everybody who was a part of making this possible in terms of financial resources to pull this off, because this is not a cheap thing to do, to bring lots of people together. So huge thanks to Neurocrine and Praxis, Longboard, Jazz, and Biogen, and then, of course, from our partners at SCN8A Spain who contributed, and the Burge family for helping pull this off, make this possible. We really appreciate your support. And could not have done it without you. All right, so let's drop that slide down and some quick final thoughts from us here. I think that much promise. We're at really, it feels like a tipping point for SCN8A. We have incredible researchers and clinicians who are committed. We have incredible patient advocacy organizations, several in the US and all around the world who are advocating and doing significant work here. We're really excited that the International SCN8A Alliance was granted a CZI grant. Rare is one, which is an $800,000 5-year grant to help us keep this work moving. It's really focused and centered on building a collaborative research network. And this kind of was the kickoff for really building that network together. And we're really looking forward to working with all of you, bringing in new people, and really trying to accelerate the science and the research and the collaborations as much as we can, because we know we've got an incredible community surrounding us here. Michael?
Yeah, no, I think that says it all pretty well. The CZI grant's going to really help us keep this wired together and moving forward. And just so everyone knows, our next step as a group of researchers, across all these different stakeholders, is to produce a written document that documents all of the things we spoke about at the meeting, what our gaps and priorities are, summarized in a sort of a codified in a working document, which will then serve as the framework for deciding what the next steps are, how we're going to reach our next goals, and how we're going to meet our funding requirements to make those goals happen.
Yeah, so lots to come. We're really excited about where we're headed and that we're doing it together. I think there are really going to be a lot of amazing opportunities for us as a community to continue to build. And I really appreciated our caregivers talking about how that families will show up if we ask them. So I think that's going to be a lot of focus too, is continuing to do a lot of work with the community about what emerging opportunities they are, either to participate in registries or biosamples, or I think a lot of those things are going to be really important coming together as we're looking to advance the work. So I'm just really excited for an opportunity to continue to work with all of our global partners. There's 10 global organizations around the world that are part of our global alliance. We're looking forward to doing more and more with TCSF to try to bring the community together and unite on some shared goals. And then, of course, working with other groups like the SCN8A community and other rare epilepsy and neurodevelopmental disorder groups to push this work forward and partner where we can to accelerate and advance this work. So I'll call it a night here. Thank you to everyone for joining us and being a part of this. We look forward to next opportunities to gather. We will do a lot more gathering in the future, more meetings coming up for both families and researchers and clinicians to carry this work forward. And of course, more opportunities to do it together, because that's really the goal of the Alliance, is to break down silos between all of these groups so that we're moving forward together. So thank you all. Appreciate it. Take care.